Good. All right. Open Source Antibiotics Series 2, Friday, January 15th. I was just saying that the recording for last time I've uploaded now, and I was looking back through it because there were an awful lot of really good suggestions for molecules. And I started to try to capture this, but I've just run out of time. And so I'm going to put this on someone. 1 second. Is that all right? So that's the meeting, yeah. So I went through I listened to the meeting again, and there were a huge number of really good suggestions here from everybody, particularly ones where we were talking about structures that we could make as feedstocks to allow, diverse syntheses of other molecules and alternative cores. So I started to work through that from these notes that I made about all the suggestions, which I've captured, but are presented raw, and we need to prioritize. And so I started sketching up a few of them there. But I'm going to have to ask someone, Dana, Ed, or somebody else, to run through the notes and just capture in image form, ideally with a string, right? Just because then that can be found, things that we think are next steps forward for chemistry. So I just ran out of time, that's all. But it's fun to do this in terms of capturing what we were talking about.
Believe it or not, for the for Chris's benzimidazole hop, I actually found the core cheaper at Sigma. It's 24 quid for 5 grams.
Okay, great. That's the kind of thing that's going to be useful. So this guy here, sorry.
Yeah.
That's great, isn't it? I mean, we can do a huge amount with that.
It's good if it's active.
No, sure, but we just need to make it one.
Yeah, that's right.
Which is very nice. So we can play with that. So that's definitely on the shopping list.
Yeah. Also, I put in the wiki a paper of related compounds based on that core.
On the benzimidazole core.
Yeah, the pyridyl substituted benzimidazole. They were doing things a bit differently. They were using some sort of click reaction. They tested them with and without a metal insert, and they found the ones without the metal were better. I couldn't get the full paper, but it's on the wiki if anyone wants to try and log in with their credentials.
Great. I missed that. If you could just link that here maybe or somewhere, just so we got it all together, in case I missed it. Or in the minutes of the last meeting or something, because I missed that you'd done that. All right, great. And then in terms of the shopping list, there were some other things here in terms of, if we wanted to try and explore this amino substitution that we were talking about last time, or something polar. That I wasn't sure. So Dana, you posted that very nice topless analysis that you did. And the suggestion there were, I don't think it suggested that we should be putting things like amines in that position from what you had. You were trying to align it with one particular, sequence in the topless analysis.
Yeah.
But I'm not sure if that would point to a justification of putting an amine there, or if we'd be going against our own analysis if we did that.
Oh boy, I don't have it at the top of my mind.
No, just
yeah, we can
I was
check.
just looking at it because there it is. So I think you were saying it was down. The progression was a bit more like this, if I remember correctly. I know this.
Oh yeah, the orange. I was thinking, right?
Yeah.
But they're not more polar, are they?
No.
No. So I think correct me if I'm wrong, but I thought your analysis was suggesting that we shouldn't be going more polar, or have I got it the wrong way around?
Yeah, no, I think that's right. Yeah, these seem to be suggesting that we have more room sterically at that position to try and put in some larger substituents.
All right. So I mean, okay. All right.
To be honest, though, A, B, C, D, and E there, none of those are particularly polar.
Yeah. I think we were talking about N-dimethyl.
I think the diethyl, which is in that orange box, might be something that we would want to go for.
Yeah.
But again, that's not going to be polar, is it?
No, not really. I mean, it's another.
It's an aniline to start with, and then
Yeah.
2 alkyl groups on it. I think somebody suggested the paracyano at some point. I seem to remember. I thought I was, that would be a more radical departure.
That's what we have then, isn't it? Isn't that the one that started this whole conversation?
I'm not sure we have that at the 4 position. I can't remember.
Oh, sorry. I think it was in the previous. Wasn't it just in the last round? I'm forgetting from here.
Oh, we did. Oh, it is active. Okay.
But this, yeah.
So
I mean, with this side chain.
Yeah, that was the compound I thought up one time. I figured probably not a bad idea to put a pseudohalogen in that location.
So I think last time we were talking about replacing this with an amine, maybe an N-dimethyl amine or something.
Right.
But I don't know if that that was a neat idea. I guess we could always try it if it's simple. But I wasn't sure if the whole point of your analysis, Dana, was to say don't do that.
Dimethylamino boronic acid should be available at Fluorochem. 4-dimethylamino phenylboronic acid should be there. I think I did a quick supplier search on the bottom of that.
Yeah, I mean, you can see that the phenol and the aniline are in the red box, so yeah, I kind of am wondering if maybe it's indicating that those are maybe not the best avenues to go down.
Yeah, and you know what? Last time we were talking about the fact that we should be being bold, and maybe these changes are smaller. So maybe this.
Yeah, they are more conservative.
Yeah, maybe this pushes down the priority list slightly in favor of exploring the other cores, perhaps.
Yeah, I think so.
So
Sorry,
speaking of which, I think that there was a sorry.
Sorry, I just wanted to ask. Can you actually put a pyridol at that position, like in that where the?
It's the nitrile with the.
Yeah. Yes. No, but I'm asking: is a pyridine tolerated at all in that position? Because I'm wondering if you could actually do like a substituted pyridine.
Yeah, I think some of the DNDi compounds had something like that, and they weren't exact matches, but they did have some activity, so that we could try that.
Okay.
Did we assimilate those in the wiki?
I think so.
Okay.
Yeah.
I'll just try and bring that up. Being slow. Everyone's streaming today. Okay.
Yeah, it'll be the different cores, so I don't think it would be in that table. Oh, but like 839.
839.
Yeah.
But that's also got the extra NH. 857 does not though. That's actually dead.
Dead.
And 854, another one, but that's also got the extra.
I wonder if you can get the boronic acid of 839, just the NH, just without the NH, put a BOH on the there and see if you can couple that. But I'm not sure if you can get that fluoropyridine boronic acid. It could be pricey.
Yeah, I mean so 4 nitrogen here, so pyridine but 4 nitrogen. So
well, yeah. I mean, I was kind of thinking about actually not putting it at the 4 position. I think we said that the like a fluoro at the 4 was possibly better.
Yeah, that was.
I'm trying to blow the cobwebs out of my brain at this point. So
no, that it was right. So I think you were looking at this compound over here, 851, and thinking.
Oh yes, right, yes. So then something to right. So it was at the 2 position. So then like a 2 fluoro 4 pyridol maybe would be something that could be kind of interesting to try.
So 851, but different fluorine positions. Sorry, is that the?
Yeah, and with the pyridine and no NH.
So it's basically 857, except for with a
Yeah.
fluoro.
Yeah.
But so 857 with the fluoro, yeah. But also, moving the N around? I mean, I guess I'm just assuming that we need something para there.
Yeah.
Yeah.
So I was thinking put the nitrogen in the 4 and then the fluorine
Yep.
in the 3 or something.
Yep.
Yeah.
I mean, if that's tolerated, it opens up a lot of different things that you could do as well.
Okay. All right. Great. We're gonna stick that on the list, right? As a specific, which is good. Sorry, I'm just trying to get back to the other picture was. So, I think the other thing as well that we needed to fix in the planning, besides buying some of the starting material and looking at whether we could get this, which was the other core that you suggested, Laurie. More difficult, less of a feedstock, right? But whether that's accessible from starting materials. And that was something else, but I forget. Is the shopping list? So I think some of the starting materials you were thinking about, Dana, for the one pot procedure and the shopping list that we might need for that.
Mm-hmm.
Were you able to order some stuff, or did I miss it?
No, I haven't ordered it yet because I'm still in the States, so that's why I haven't done it.
Yeah, no, okay. Are you happy to think about the things we need to get in?
Yes, I'll make a shopping list, and then once I know what I'm doing, I'll order them so that I can have them when I come back.
Great.
Did we ever make anything with you've got R2N there as a position at the 4 position, but what about making it put a CH2N so it actually is basic?
Don't think we have anything of that kind.
No, I don't think so.
Yeah, I'm still a bit concerned that these are all quite lipophilic. So if you could get a ionizable group in there, I think it would be quite nice.
Nothing like that there, I think. Right, so benzyl amine. In the 4 position, all right. Yeah. Sorry, I just got back to this thing again. Yeah, and the stock of this guy. So I think that there was some. So Ed had just done the scale up of this guy for the HIFA stuff. So he got 500 mgs or something of this guy, and so he wanted to play with chemistry, like for example the idea that Chris was talking about with alkylating the nitrogen and reducing this to generate saturated analogs. If we wanted to play with that, we can.
Cool.
Which is good. But the idea people had of playing with this position would require a bunch of this kind of compound to be made.
Yeah.
So again, that's just on the shopping list, and just to make sure that we've got stuff that we can generate that. And then, as I mentioned, that there's a huge amount of, let me just too close in. There's a huge amount of other stuff in here which we were mentioning, and we just got to put it in some order list. That's all, so that we have a clear set of targets. Trying to mix some of the SAR stuff that we'll need for the paper with more bold suggestions. All right. I mean, the other things here are fairly quick, I guess. So there's been no chemistry updates because, of course, we've been away, and the lab is still closed-ish at the moment until Ed and Dana can get back in. So we haven't got a lot of chemistry stuff to do, but of course we can chase the cytotox, which I'm guessing is also slightly delayed, but we can keep asking.
I've corresponded with them, and let's see. So emailed Reno last Friday. He said, I'll continue working on your compounds next week when Alex has come in after her self-isolation. So they're also having COVID problems. But we can just keep it, on their radar, and I think they are planning to pick it up soon.
Fine. Okay. Awesome. And there was just this note about the tox data on your compounds, Laurie, and what we learned from that. Am I behind the times there? Is this already done, or have we? Because I still don't remember thinking about that. I guess it's somewhere, right?
I'm pretty sure I sent it to you, Dana.
Fine. Okay.
I'm pretty sure you did too, but I can't remember if I put it on GitHub or not.
Sorry for being about Striver.
Okay.
Okay, fine. If we could just find out, dig it out, put it up there, and then analyze what it says, that would be helpful. Yep. I think. And then the HIFE results came in. So they've been busy, and they've got these nice results, which I guess is, I mean, it's not big news yet. It's just that things are proceeding according to plan. In the sense that they've done their big screen and they've found some nice results that give them a decent amount of a pretty simple oxygenated, mono-oxygenated hydroxylated, I should say, metabolite, which they think that they can scale up and send us. So the plan is, it seems they're quite busy this month, but they were planning to do this in February. The plan is that they are going to do that scale up after playing around with a few different conditions, and send us the crude material, which we then purify and characterize by NMR. So that's the deal. It's they're gonna carry out that scale up for us, and then we do everything else after that, which is really good. So I mean, I was hoping exactly for this, that they found something that was looking decent and should allow us to find out what we've got. 2 monohydroxylated metabolites, which is the thing. Those are the things that we get from them. That's good. I mean, something, and we'll see what they say.
Have you ever looked at your compounds in Metasite?
I think we did, but I forget who did that. Why
We've looked at them in a few predicted programs.
Okay.
I think Alvaro ran Metasite on his compounds, but then that I think is licensed. So then we were looking at, yeah. So then, but this is, I think, the only one that we have the Metasite data for. And the other ones like Smart SIP and other things.
Okay.
So we wait, but it's good that they're progressing and they're still active in the lab, which is great.
Mm-hmm.
And then the other thing, which was I guess the main item for us, particularly during lockdown, is the mechanism of action stuff. So Dana posted very nice analysis of, which of course I can't find, of this stuff now, which I will dig up. So wait, why can't I find it? Amazing.
Oh, because we started it a long time ago, so it's like issue 15 or something.
Is it? Okay, fine.
Yeah.
Okay, man. Oh, there. Yeah, great. So yeah, I mean, just so we can understand the experiment, the data. Dana, do you want to talk us through this? I just read it.
Yeah. Okay. So this paper that I pulled this image from is Lee is an author on it, and so it's sort of specific to the experiment that they were doing, but I think the same experimental design. So they have the beads, so they have kinase inhibitors that are mobilized on the beads, so that's basically the solid phase. Then they either incubate the MRSA cells with DMSO or our compounds. So we sent them an active and an inactive. So they did both of those. And then, so that's your mobile phase. And basically the kinase inhibitors on the beads will pull down any kinases that they bind to. So if you see decreased binding in the presence of compound, then that's an indication that your compound is out competing whatever the inhibitor is on the bead that would be pulling down the protein. So that's the most straightforward way to interpret the results, I think. And so then the data that they sent us was basically a list of proteins. I'm not sure whether they're all kinases or not. But anyway, and then they've calculated this log2 fold change, so it's relative to DMSO for the active compound and the inactive one. So does that make sense?
Yeah, and I guess that's the thing, isn't it? You don't need to worry about what the binding interaction is, or whether or not you're getting good interaction between the small molecule and the protein. All you're doing is seeing if it's a change when you add your compound in.
Yeah. So it's basically a proteomics sort of profiling experiment. So the ones with a negative fold change mean that the protein is, I don't know, less abundant in the presence of compounds, and then a positive fold change means that it's more abundant. So there were quite a few with a positive fold change. So one of the questions that I had for them would be like, what exactly would the mechanism be? So where your compound would be increasing the amount of protein that would be pulled down. I mean, I guess maybe that would tell you that this compound may be hitting a protein that's upstream and upregulating this target that then shows increased abundance. But I think I'd like to just hear what they have to say about that for sure.
Yeah, absolutely. So what I was going to suggest is that I ask them to come along to this meeting next week.
Yeah, that'd be good.
And talk about it, because now you've done this analysis, we all understand what's going on. That's great. I mean, I don't know what this means.
Yes.
Right. So then the other thing I'm not clear on, I think that these are MRSA kinases, but we need to know how they've identified them. I mean, I know it's by mass spec, but is it related to the B that they've bound to, or have they done like MSMS? That I don't know. And we need to figure out whether, so if these are human kinases, it's going to be a bit more complicated. Or whether they've actually gone through and looked at, you know, what's known about MRSA and the kinases that it expresses, and done it based on that.
Yeah. Okay. So I mean, I assume that this is all MRSA.
I think it is, because there's a sheet in their Excel document that has the species in there. But so I mean, that would be just nice to get that confirmed by them, so we're not, you know, wondering a bit. And then I think we need to find out, you know, what's known about these targets and whether it's reasonable to expect that that would be the mechanism.
Yeah. Right. Okay. I'm just thinking about whether that's true, if they're all. Yeah, I don't know actually. I don't know the extent to which these are immersive versus human. Does anybody else have experience with this?
Slightly. I know the four-letter, the lowercase 3 with a capital A, that four-letter scheme is used in bacteriology and biotech. When you're engineering bacteria, that's usually how you name the genes. So these are likely the bacterial ones. If you look them up on Uniprot, you should get something.
No, sure. I'm trying to work out from the experiment whether we're talking about what kind of targets we're talking about here. That's all.
Well, yeah. I mean, I think it would have to be MRSA, right? Because that's the cell lysate that they used.
But whether they're unique to MRSA, I guess, is the question.
Yeah.
Some of these don't look like kinases too. Some of these could be other enzymes. They most likely pulled down everything.
I guess I'm just thinking that because I think the request to use MRSA was for them unusual. So I'm just wondering how they know all these proteins already, how they're so confident that.
Yeah, that was my question. I mean, maybe I think. I mean, I don't know. I guess there must be like a database or something that they would use to, if they're identifying these proteins by mass spec. But I think we need to definitely find out like how exactly they've assigned.
I know that there is a database regarding these proteins. I need to check again the name, but there is a database in which like proteomics analyses are detected.
Yeah, but is it for MRSA or is it for humans? Do you know?
Is for I think everything.
Okay. Okay.
Because I guess I know the group that works there at UNC has a huge amount of expertise on human kinases. So I
Yeah.
guess I'm just thinking how it is. Maybe it's by analogy. I don't know. So I think that's going to be our first question, isn't it? To make sure that we're dealing with those. Then of course the question is, well, all right, if we're talking about MRSA targets, then what are we going to do? If we have a short list of potentials, can we access those in order to confirm the results?
Yeah.
We can always try docking first to those MRSA targets.
Maybe, but I mean, I guess
you can only really do that if known crystal structures, can't we?
Yeah. Yeah.
Maybe some for a couple of these proteins.
So I mean, ideally, we'd have a shorter list than that, right?
Right. Well,
yeah.
That if we went through and found out whether these have any assigned functions, there could be some that we could eliminate based on that.
Yeah.
That shouldn't be too hard to do. That's a quick search through Google, through UniProt.
But then there's also a question about if any of these are functionally connected, then that could be evidence that we are hitting something.
Yeah.
If other potential binders. Well, actually, no, I'm not sure that's true. Actually, I'm not sure that's true because this is a lysate experiment, isn't it?
I can build a spreadsheet of UniProt entries.
Yeah, if the codes tell us something, that would be useful. Sure, and then just dump it somewhere. That would be useful.
Yeah, those I reckon. Yeah, this was something I a little bit familiar with because I know someone who used to do this. Not this, but just engineer bacteria in general. But yeah, they use these three lowercase followed by a capital A to denote a bacterial gene. Sure. So these are likely bacterial genes.
All right, but I think we can answer all these things next week when we get them on. We have the team themselves on speed dial, so we can just get them in and they can explain the whole thing, which is really good. And so I think it's given the number of potential proteins in that spreadsheet, the fact that there are this small number of those that are relevant is quite good news at the outset. I'm just saying ultimately, experimentally, hopefully we can whittle it down even more. But great that it's been done. I don't know to what extent they're available for follow-up experiments, but I suspect that they will be. So that's good. All right, so I think we'll dominate the next meeting with that. That'll be pretty nice. All right, and I think all the other things were again just actions for people to deal with. The only thing I was just going to highlight, I think, was the molecule donations terms. I tweaked a little bit whilst I was thinking about it over the break, and if they're okay, because I haven't changed anything, I just sort of grouped them differently to try and make them a little bit easier to understand. So there's a new one posted here a week and a half ago, where I just tried to give them headings and try to group them together. Then if everyone's okay with those, then those just need to go onto the form, Dana, that you had already generated.
Oh, I think I already did that.
Okay, fantastic, great.
And I think I've updated the landing page too.
Fantastic, great. All right, that's great. But nothing new there, just a different way of expressing it.
Mm-hmm.
And then the rest of it was as per. So there's a bunch of action items which we haven't done. Christmas is one of those times when you get done far less than you think you're going to get done. Even in a lockdown Christmas, I didn't get all these things done. So lots of things there for us to do. Was there anything else anyone was thinking about or wanted to talk about before we wrap up?
If you can log in to get the benzimidazole paper, we can all look at it together. Okay. It's on the wiki. It's in related compounds.
Oh, right. You mean the landscape thing? Yeah, there. You know where it is?
Bottommost.
Oh, this guy. Okay. We may have to just, if everyone can talk amongst themselves, I can probably do this. Well, actually, you know what? We can do this offline. What we might be able to do is share it around us a little bit and then have a think about it rather than you watching me entertainingly log in through ECL's portal. But that's great that you've done that. So that's an item we can look at and think about for next time. Just conscious of people's time, that's all.
Okay, that sounds good.
Yep, all right, great. So next week we'll focus on the other stuff, but until then, if there's nothing else, have a great week. Thanks for coming.
All right. Thank you very much. Bye.
See you.
Bye.
Bye.